Okay, so what we're going to do is review this standard, which is using a model. You're supposed to use a model to how photosynthesis transforms light energy into stored chemical energy. We've been using clay models to help act this out, but we also, there are so many different models out there, like two-dimensional paper graphic models as well. And it's important that we can use models. So with this one, again, any model is obviously a lot more simplified than what is actually occurring in nature. And we use different things to represent different components or different actions. But ultimately, if I give you a blank model like this, the end goal is that we can end up like this. And again, this looks super complicated, and there's lots of things. And again, sometimes when we simplify things, we make them inaccurate, but we're trying to get some big points across as far as how do we get from light energy and water and carbon dioxide ultimately to energy stored in glucose and producing oxygen as a byproduct as well. So in order to understand this better, we've gone through it in clay, but in order to understand this better, we're also going to go through it one step at a time here on this, uh, graphical model. So before we get there, let's make sure we understand all of the players or the components involved here. So photosynthetic organisms aren't just trees and plants. 50 to 70% of all of the oxygen or photosynthesis that takes place actually happens in the ocean with microscopic organisms, little protista, little algae, little cyanobacteria. But all of this works through these chloroplasts that exist within cells. So we have these structures called chloroplasts. They're typically green because of the chlorophyll, which is absorbing red and blue wavelengths, but reflecting green and yellow wavelengths, and therefore plants look green. But we also have other things involved here. And overall, we have this overall structure called the chloroplast, but within the chloroplast, we have the light-dependent reactions happening with thylakoids. And we're just showing one big thylakoid here, when actually they're stacked up and they're much smaller than what we're showing here. But again, in order to understand things, we try to take complex ideas and make them simpler. As we do that, we often make things somewhat inaccurate. But we have water, we have light, we have protons, which are the little gray dots. We've got proton pumps in between the chlorophyll. We've got this enzyme that helps turn NADP plus into NADPH. We have ATP synthase, a little channel with an enzyme attached to it. We have ADP, a free phosphate, Rubisco enzyme in the stroma, which is the outside of the thylakoid. We have carbon dioxide. So from these components, we're going to end up showing how we can take energy from light and turn it into energy in the bonds of carbohydrates like glucose. So again, you start with this, and we're going to go through it one step at a time here. And the very first thing, and again, I want you to kind of keep in mind here, we're going to kind of read left to right and walk through the light dependent and then the Calvin cycle. So the very first thing we've got going on is water. Water is going to come in to the thylakoid. It's going to be broken down into oxygen, which is going to leave, protons, which will stay in the thylakoid, and electrons, which will get transferred to that first chlorophyll right here. And again, the electrons are meant to be the lightning bolts, and the protons are the little dots here. Protons are sometimes called hydrogen ions because together the electron and the proton here makes hydrogen, so it's H2O. Our second step after water is broken down is light is absorbed by that chlorophyll and it energizes the electron. So again, we're reading left to right. We get our water broken down, our light comes in, and this is step 2. The electrons get energized. The electrons then power the pumps. So for step 3, we're moving these electrons over to the pump, and that allows for active transport of the protons into the thylakoid. So it's going from low concentration of protons to high concentration of protons, therefore some type of energy needs to be used to help actively transport those across this thylakoid membrane. Step 5, we need to re-energize the electron. So light comes in, is absorbed by the chlorophyll, re-energizes the electrons, and then those electrons get carried to the Calvin cycle. So this NADP plus, the truck waiting here at the loading dock, this enzyme, becomes NADPH. So it picks the electrons off of the electron transport chain, gathers up some hydrogen or some protons. So ultimately, it's taking these high energy electrons, which just got energized by the light, and we're going to carry these over towards the Calvin cycle here, where we can add them to the carbon dioxide in order to store energy into the bonds of glucose. Our 7th step here is to create ATP, and that happens as the protons want to get out of this thylakoid, but the only escape route is through ATP synthase. So they go through this channel, and as that happens, they help this enzyme here add the phosphate to ADP to become ATP. And I put the little star here by that last bond to represent that energy is mostly stored in that last bond. Now we're jumping over to the Calvin cycle in the stroma. So over here, all the carbon dioxide is going to come in, and the Calvin cycle, again, we're super simplifying this to make it easier to understand. It's a super complex cycle, multiple rounds through, changing the number of carbons, and many enzymes are involved. But our big idea here is that we are taking carbon dioxide, we're using this Rubisco enzyme, which is the most abundant protein on the planet, and we're taking the hydrogen, the electrons and protons off of the NADPH, we're taking the energy from ATP, and we are creating glucose sugar, and that's what this is meant to represent. This 6-carbon sugar with some hydrogen and lots of energy stored in it. Okay, so again, if we are given this blank template, this model, we need to illustrate how photosynthesis transforms light energy into stored chemical energy, and this is how it would look as a fully completed process. Step 1, water is broken down. Step 2, light energizes the electrons. Step 3, again, we're just showing the pathway of the electrons here along the electron transport chain in the light-dependent reactions. Step 3, electrons power the pumps. Step 4, protons get pumped. Step 5, light re-energizes those electrons. Step 6, NADPH is created as the electrons and protons are picked up. Step 7, ATP is created as the protons diffuse out of the thylakoid through ATP synthase. Step 8, carbon dioxide comes in to the Rubisco enzyme. It takes the electrons and protons, the hydrogen, off of the NADPH, takes the ATP, and we end up with glucose sugar. And that is what I'm looking for on your assessment. So if we just go fast here, 1, water is broken down. Step 2, light energizes the electrons power the pumps, the proteins get pumped, light re-energizes the electrons, we get NADPH as it picks up the electrons, we get ATP as the protons go out of the thylakoid through ATP synthase, and finally we get glucose as carbon dioxide comes in with the help of enzymes, picks up electrons and protons and energy from ATP, and we end up with glucose. So that is photosynthesis. Again, if we look at it from a chemical formula standpoint, we have carbon dioxide and we have water. That's what we start with as reactants on this side of the chemical formula. What we get as products are oxygen and glucose. So if we're looking at this from a chemical reaction standpoint, we balance the chemical formula. We start with carbon dioxide and water as reactants, and we produce C6H12O6, which is glucose, and oxygen gas. That is that.